So good afternoon, everyone. The more observant amongst you might notice that I'm not Sakis, who unfortunately is not well and kindly emailed me to say he doesn't want to spread his germs and therefore he'll stay away and sent his apologies, and so I will take over in his stead. So welcome to our 2017 seminar series. This year our seminar series has got a special title. We call it the Young Academics Forum. And the aim is, first of all, to provide a forum for our own young academic staff to invite to the department other young academics from around the world and to build long-lasting relationships. So the idea is for us all to become acquainted with some of the rising stars in chemical engineering around the world, expose them, of course, to what we're doing in research and teaching, meeting colleagues, finding out what we do in our research groups, building an understanding of the breadth and depth of our department, while also learning about what goes on in their own institutions. It's a unique opportunity for us to be informed about their success stories, learn about their ongoing research, and be inspired by their ideas and vision. So in that spirit, today's speaker is Dr. Robert Grass. He is from the Department of Chemistry and Applied Biosciences at ETH. He studied chemical engineering at ETH and had a short stay at Case Western Reserve in Cleveland in the United States. It says here in 2,993, but I imagine it's a typo. He returned to ETH to pursue his PhD studies, and he's currently a lecturer in ETH in the group of Professor Stark. He works in the teaching side on the topic of process control. So in his relatively short career, he has actually written over 100 research papers, produced 8 patents, and 3 book chapters. He founded a company in 2007 called TurboBeads, and this makes chemically functionalized magnetic materials, and they are commercially available through Sigma Aldrich. His research is at the interface of nanoparticles, surfaces, and biochemistry, where he's pioneered the use of carbon-coated metallic nanoparticles for separation processes. More recently, and we'll hear a bit more about this later on, he's been looking at synthesis processes of bioanalytical applications of functionalized particles. And a current focus is on using encapsulated DNA as an information carrier in digital information storage, product barcoding, and material labeling. So please join me in warmly welcoming Professor Robert Grass, who's going to share with us his experience and research work on the topic of sending and receiving information by chemistry. So over to you, Robert.
Thank you very much. Thank you very much for the very kind introduction. Thank you very much for inviting me. Ronnie, thanks very much for initiating that I was invited. Thank you for showing up. I want to talk about information. And as chemists, you might ask, well, why do we want to talk about information? Because what we chemists or chemical engineers, we make physical things that matter, like the polymer in my laser pointer. We make the basis of the drugs which heal our illnesses, and these industries make tremendous amounts of money out of doing that. If we then look at other companies, especially in recent times, we see, well, there are other companies which make nearly as much money or even more money. They make even more profit per dollar of revenue. And these companies don't sell anything different than information. So obviously there's value in information. And what we want to make sure that our field isn't let out, that we see what can we do with chemistry or chemical engineering in this world where information is so extremely important. Now if we think in chemistry and information, you very quickly think, well, analytical chemistry, that's information, where we take something from a reaction mixture, a sample from nature, we put it into a machine, and it will yield some kind of information. But what I want to do is more than that, not just gather information on something that happened, but I want to play with information. I want to manipulate information or send information to somebody else. And then we might want to think, well, if I want to do that with chemistry, which molecule would be the best suited to do that, and why would we choose that molecule? And I believe DNA is the molecule which is extremely well suited for that purpose. And I'll quickly tell you why I think that's so well suited. DNA is unique from its analytical chemistry point of view. Because if I have DNA, let's say 100 base pair of DNA, I can detect one single molecule of DNA with a relatively simple apparatus. That's unique in the chemical world. Usually to detect atoms or molecules, we need millions of the same molecule to be able to detect it, and often the infrastructure we use for it is pretty tremendous. Also, I can not only detect the sequence of a DNA molecule, the presence of a DNA molecule, I can also read the full sequence, so the base pair sequence of that molecule, yielding me about 200 bits, not about, yielding me exactly 200 bits of information I receive when I read that molecule. And that's from just 0.1 attogram of matter. So that's nanogram, picogram, femtogram, attogram, 0.1 attogram. And I can still not only know presence, but also sequence. And we all know nature does it best, and that's what nature does. Nature uses DNA to store our genetic information. All our functions are stored in DNA. Our genome, 3 billion base pairs, few meters long of DNA, is equivalent to about 1 gigabyte of information. And every cell of our body has that gigabyte of information, and we have like 1 trillion cells. So you can imagine the tremendous amount of information stored as DNA in our body. Nature also uses RNA to send information and to then generate function by translation. Now, we want to use this in an information world. We might want to compare it to what we are better known in the information world, binary code, and we can have a genetic code. If we look at the two, they're extremely similar, where the binary code, we store information by a sequence of zeros and ones, and in DNA, we store information, or nature stores information, by having a sequence of these A, C, T, and G, 4 different possibilities of the base. So they're extremely similar. Also, if we look when they were developed or when they were recognized as such, in 1937, Claude Shannon is seen as, let's say, the father of using binary in computing, and just about 15 years later, the, let's say, way DNA works or encodes for information was discovered famously by Watson and Crick. So not only nature can use DNA, we can also synthesize DNA at will. We kind of have to imagine that like Lego, where we can take one base pair and stack it to the next and build a specific sequence of interest. Of course, we don't do that as Lego. There's real chemistry doing that, where on a solid phase and protection group chemistry, we can introduce one base after next, and through every cycle we do here, we can introduce another base of information free at will. And we can not only do that one sequence at a time, but that can be done on a chip, where on such a chip you have thousands of these spots, and electrochemically the sequence of the DNA can be directed by saying which spot shall react with which base. And with that, there's really technology out there to generate and synthesize large amounts of DNA with given specific sequences. There's a whole industry in the biochemical world using this technology. That's writing information in DNA. We can also read information in DNA, most famously next generation sequencing, or mostly Illumina sequencing, where similarly on flat surfaces, DNA is read by using dyes, bases attached to a dye, and then we can take photographs of the growing of the DNA sequence and then recalculate the individual sequence. So you can measure millions of DNA sequences at the same time using this Illumina sequencing. That's also reflected in the cost of sequencing DNA analysis. So Human Genome Project costed about $100 million to get one human genome. That's not so long ago. Today, so we are 17, you can get your genome analyzed for $1,000 just a few years later. And you can see that this decrease in cost is significantly faster than Moore's law and shows this enormous effort that has been put into DNA analysis in the field of biomedical engineering. So we can write DNA, we can read DNA. DNA is also said that it's extremely stable, which is an important property if we want to use it as an information carrier. This bone is 700,000 years old. It was found somewhere near Alaska where it's really cold. And the genome of this horse, of whose bone this comes from, was reread a few years ago. So that's like the past 700,000 years ago, sending us 0.5 gigabyte of information to modern times, which is pretty impressive in this long time period that this DNA has been stable. That's, however, a problem with DNA stability. DNA isn't so stable, isn't as stable as we would like to imagine. Because we know in our body we have thousands of DNA errors happening every day. These three gentlemen received a Nobel laureate for analyzing that and understanding that. And also in the environment, if you have DNA lying around in the environment, it doesn't last long. Even in the lab, it'll just last for about a year at room temperature. So somehow there's a conflict between this bone and the, let's say, DNA just decaying very rapidly. And the solution to that, and what's, let's say, falsely accredited to the DNA in this bone example, is that stability doesn't come by DNA alone, but it comes by the bone. Because the DNA is encapsulated in the bone, and the bone gives it its stability. So it's like in a fossil, like if you know the movie Jurassic Park, where you have this fly in an amber fossil and the DNA is extracted. That's not so far away. There has a few problems. DNA is not stable in amber, for example, and amber is also much too old to still contain any DNA. But in bone, encapsulated DNA is extremely stable. So in order to utilize DNA as an information carrier, we have to generate something like this fossil. We have to find a way to put DNA into something so that it's more stable than it is just free in solution. And so we developed a technology which we call a synthetic fossil, where we encapsulate the DNA specifically in an inorganic material. We didn't choose calcium phosphate as the bone, because calcium phosphates aren't very stable by themselves against dissolution in specific chemical conditions. We chose silica, glass. We wanted to encapsulate DNA in glass. For that, we first use this glass nanoparticle, and we give it a positive surface charge by some surface chemistry. We then add DNA, and DNA, by its negatively charged nature, will just adsorb on the surface of the positively charged particle. So we have DNA on the surface of this particle. The outer side will be negatively charged because of the charge that the phosphate groups of the DNA carry. Then we use this molecule, which on the one side is a cation, which will bind to the DNA. And on the other side is like a silica precursor molecule, with which we can then induce the growth of silica on that side. We then add more of another silica precursor, well-known silica precursor, which will polymerize on the outer surface, and we will grow like a glass silica shell around the DNA molecule which deposited on the original particle. We can imagine that it looks like that. You have the DNA kind of suspended between these cations, and all of that is in a solid silica matrix. These are images of these particles. You could see, perhaps, this is the original particle, and then you have a new layer which is grown on top of the DNA. You could speculate, if you wanted, that there's like a gap where the DNA might be. But if you ever did electron microscopy, you always see things you like in electron microscopy. So we are not so sure if the DNA is really there. So we had to find a test, a chemical test, to find out if the DNA is really inside the particle and not outside or somewhere where we don't want to have it. And for that, we decided to use a radical treatment, where if we take DNA and we treat it with these ROS radicals, which we induce in solution, DNA is just destroyed immediately in the presence of these radicals. However, if the DNA is encapsulated in my particles, independent of the duration of this radical treatment, the DNA doesn't decay. proving that the DNA has to be inside something solid where these radicals can't get in. The radicals are extremely small, proving that I more have a covered, closed shell of material surrounding my DNA. So you might say, well, yes, we can put DNA into this glass particle, but a big problem is we have to analyze the DNA. So we have to get it out. So how do we get the DNA out of this particle in which it's protected from hydrolysis and also temperature? And the solution to that is found in inorganic chemistry. If you want to dissolve glass, what do you use? Hydrofluoric acid. Glass dissolves selectively in hydrofluoric acid. But most people are afraid of hydrofluoric acid and additionally believe that hydrofluoric acid is a very aggressive chemical and will certainly destroy DNA. Both is not true or not completely true. Hydrofluoric acid is extremely dangerous. However, hydrofluoric acid is also in your toothpaste as ammonium fluoride or sodium fluoride or in some buffered form at low concentration. And so we can use such a low concentrated formulation of hydrogen fluoride to selectively dissolve the silica, and what stays back is just the DNA. And all our downstream processing or analysis of DNA also doesn't care about these species. The proteins are stable, we can do PCR sequencing, everything in the presence of these ions. They're not as, let's say, acidic as we would expect by just thinking about hydrofluoric acid. So let's look at stability, real temperature stability. This is the stability of DNA in water. So you see at 20 degrees you get about part of a year in stability. If we then put the DNA into our silica particles, we see a significant increase in its thermal stability. So we have to simulate the decay by heating it up to high temperatures. There's no way we can wait for hundreds of years to measure the stability. But you can see this enormous increase in stability. And also, if we dare to extrapolate it, I know it's extremely dangerous, but it follows activation energy rules. We can kind of predict that it's almost as stable as DNA is found in the solid bone, which is the same. It's DNA encapsulated in a solid matrix and gives a specific stability. So now we formed these particles, which are more or less indestructible in environment. They can survive for extreme long time durations. And every particle has about 1,000 DNA strands of 100 base pairs, so you can have 200,000 bits of information in such a particle. And a question would then be, can we use this in a data storage application? And you might ask, well, we have excellent data storage. We have hard disks, floppy disks, memory sticks. They all work. Why do you want to use something as exotic as DNA as an information storage device? And the problem is shown here is the timeline of storage duration that these devices bring. So we've significantly increased the storage density in, let's say, making better and better technologies, but we have also significantly decreased the stability of the information. We forget that information on our memory stick is probably not there in a few years, and information on our hard disk or DVD won't be there in 10 years. That's a problem. We are going to lose the information we have, and we have enormous amounts of information. If you ask companies like Facebook or Google or Amazon, the amount of information they have, the amount of data centers they have to build to sustain that information is gigantic. So there is a need of, let's say, further increasing density plus having a higher stability of information. So then you could devise an experiment. Let's take some information. We somehow translate it to a code of ACTNG. We'll get to that in a moment. We have the DNA synthesized. We encapsulate it in our particles. Heat treat the particles to simulate damage. We then, after that, release the DNA, sequence it, translate it back, and hopefully get the same information as we had originally proven that it can be used as a data storage device. one problem is nothing's perfect. So reading and writing is not perfect, and doing thermal damage simulation, we will have some damage. Even if damage is much slower than in the case without the protection, there'll always be some kind of thermal damage in the material. So we have to find a way of getting around that problem of how do we handle that damage? What can we do with that? Because we have, as I said, write errors, reading errors. It's possible that we lose information. And I was lucky enough to meet Reinhard Heckel. He was a PhD student in communication theory, and he told me, well, that's not a new problem. Any storage device has that problem. We're just not aware of it. Any storage device makes errors all of the time. And what industry or what that world has devised is error correction schemes. So you add additional amount of information to the information you want to store in terms of redundancy, so that you can calculate out specific errors that have happened during storage. The best way to explain that, I won't go into the technical details, but just to give you an idea why that's important and where you've seen that is that if you have a DVD and you scratch it, the information under the scratch is lost. Still the DVD works, unless you've severely scratched it, but in normal case, it still works. And it only works because there's redundant information somewhere else on the disk with which you can compute the missing information. That's done in, let's say, complicated mathematic technologies. I don't have time to go into that. It was developed in communication to satellites, where you can imagine that if you communicate with a satellite, you lose a lot of information on the way there and on the way back. So you have to find very elegant ways of introducing redundancy, so adding the information again. But we don't do it by physically adding it again, let's say writing information, but we write information and we add a few bits of information with which we can calculate mathematically we can correct any error in the information word. And so what we did with Reinhard was to implement this idea of error correction into DNA storage. So in this translation from binary to DNA, we encoded a code which added about 20% of redundancy, so more information than we wanted to store, and then also calculated that back on the reverse. And I'll just show you on the reverse to give you an idea how that works. So in DNA sequencing, so we have our DNA which we bought in a pool, in a liquid vial, in a few microliters of water. And we then, for sequencing, we more or less statistically pull sequences from there and we read them. And these individual sequences, they will have errors at specific points. But we have a code which can decode that and remove the errors to get back to original sequences. So there's a little redundancy added at the end with which we can recalculate out any errors. Then we have an index at the beginning because we have to arrange our information in its original order, because otherwise it's just random sequences of information. So we have to arrange them to the beginning and so on. And so we use that index to arrange that information. And it's possible that some information was lost, some sequences were lost completely, either by damage, by double strand breaks, by reading that they were at the end of the chip or they didn't read perfectly. So we will have errors in completely missing sequences. And for that, we have this added redundancy here. So we have synthesized more sequences than we would have to had. We would need to decode the information, which is this redundancy block here. And with that block, we can recalculate the gaps in our original information and hopefully get back all the information we stored. If we look at the results of that, so that's it. DNA as we bought it, you can see it has about 50% error level. That means every sequence has so every second sequence has an error. But if we use our coding strategy by inner and outer coding, these two ways of decoding, we get back the information without any error. We can perfectly recover the information, either original or artificially decayed to 1 or 4 half-lives of DNA. 1 or 4 half-life of DNA in our case is about 500 and 2,000 years of storage of that information. So we've proven we can store information in DNA, store it for 2,000 years, and recover it without any single error. So we can prove that these two are essentially the same. And that we can, let's say, close this gap back here and have 1,000 years, more than 1,000 years of stability. And due to the enormous density we get in storing information in DNA, because every base is 2 bits, and Avogadro number you have 6 to the 23 bases per 660 grams per mole. So enormous amount of information. You can get enormous data densities, even if we calculate that we lose a lot of density by just having silica. Most of our material is just silica, protecting the DNA, and a small part of it is DNA. But even then, we can get these enormous data densities. So as a scientist, I will tell you the future of data storage, of archival data storage. It'll never be in your computer. Of archival data storage is in DNA. As an engineer, I have to tell you, please back up your data, because it'll take at least 20 years until we can get that on a, let's say, economic scale. Because at the moment, it costs $2,000 per megabyte. I don't know, 2,000 megabytes is perhaps an image. Don't know who has an image where he would be prepared to spend $2,000 to store it. So we're still far away from that, from an, let's say, engineering aspect. So we might think, well, what other things can we do with this information in here? Do we always need these enormous amounts of storing gigabytes or terabytes of information? Perhaps having a smaller amount of information would be useful as well. So we came up with the idea of using this as a tracer, as a barcode, which we could add to a product, because you have a protecting envelope. Inside you have a DNA sequence which encodes like a barcode, 200 bits. It's already probably more than you have in most barcodes on product packaging. The difference with this barcode is not on a product packaging. You can put it into the product, and it can be never removed from the product. Wherever you have product, you have information imprinted in that material by DNA. So we could think of, let's say, product paternity testing, if you want it, that you put them into a polymer, you process the polymer to make a device, sell something, and you could always go back and read, well, who made the polymer, where does it come from, information you put in by using DNA encoding. That would be interesting for a device such as a knife, but I think it would be even more interesting, perhaps also scary, if your food was traced, if you know where your food comes from, because you're prepared to pay a lot more for pasta that comes from Italy than pasta that comes from Brazil, or I don't know, somewhere else. So there's an added value if you know where your foodstuff comes from. At large, you're prepared to pay double or 4 times the amount if you know who made your product. So there's a need for that. One big field of counterfeit products is olive oil. I don't know, 50% of the olive oil you buy is not extra virgin olive oil, although it says. So it was for us an example to show that we can use these particles, add them to olive oil or to bergamot oil or to gasoline, and show at concentrations of 1 microgram per liter. 1 microgram of these particles per liter of oil is sufficient to not only identify the sequence of the DNA, but also to quantify if it was diluted or if it was otherwise handled in a way it shouldn't have been. I work in Switzerland. We make cheese. So another experiment by a student of mine was to add them to milk, show that we can process the milk to yogurt and cheese, then remove the particles from the cheese, release the DNA, and by polymerase chain reaction, we can show that through the processing, through various processing steps, we can still go back and get a quantitative measure of where the product came from. We also have some biologists in the lab. When we showed them our traces and the possibilities, they said, well, we could do a really neat experiment where we trace foodstuff in a food chain. Let's say we take some flies and we feed them to food that has the particles inside. Can we measure which flies have eaten which food? And we can see that we can really detect the particles in the fly. So we have to digest the flies, because in the gut we find the particle and the DNA signature of the foodstuff we've given to the fly. We can do that for flies, we can do that for mealworms. Can then even go a step further and find some spiders which will eat the flies. And then we can detect which spider has eaten which fly, which has eaten which original food. And it's like first experiment of showing that we can do like ecological tracing, especially in insects, in understanding how insects travel, how far they go, predatory patterns. I think it's a really interesting tool in getting an experimental picture of these things. So we mark flowers with different particles, with different codes, white flowers and red flowers, and see if the flies have a preference for the white flower or for the red flower, which indeed they do. You see that you see a very strong preference for red flowered for this fly. When working with these things, you start thinking about, well, what's our detection limit of this method? How low can we go? We said we can do 1 ppb, 1 part per billion of particles. But can we go lower? Can we go to a level where we only detect one single particle in our solution? Well, problem with detecting one single particle comes to the idea of a digital measurement of particles. Because if you have, in PCR, you often have 100 wells, and you put in different samples. And if you think, well, if my sample is really dilute, and I put it into 100 wells, and on average I have half a particle per well, some particles will amplify and give me signal, and some won't. And by taking into account statistics, Poisson statistics of the distribution of my half particle on average per well, I can really go and count particles. I can say, well, this one which amplified here, I had a particle. In this volume which didn't amplify, I didn't have a particle, and so on. So I can go and count the number of particles in my sample one by one. And I think that's really unique. If you come of a world of particle analytics or tracing or of nanoparticles, being able to measure and count individual particles, we have a really unique technology. We've shown here we can measure these 137 nanometer particles. We can even measure much smaller particles. And for us, we kind of said, well, it's like counting sheep, counting individual particles. You really have a discrete measure. It's not like a device which gives you 2,347.5, but you're really measuring one particle after another, one by one. Now I've shown you that we can detect one particle. We can use it as a tracer. But can we do more than just that? Can this technology, can we measure something? Can we go and think of our particle as a measurement device, which we put somewhere, we measure something, we store that information that we measure, and later when we recover the particle, we are able to read out the information which we have measured. And playing with this idea and thinking about it, I told you before, we know a lot about thermal stability of DNA or decay patterns of DNA. So you could take the idea and say, well, I can have some kind of a stress, and as a response of that stress, my DNA will decay. If I know how DNA decays, I can use that as a measure for that stress. And so I want to show you two examples of that. First, stress is temperature. We know that RNA decays is more sensitive to thermal stress than DNA. So we put RNA and DNA into the same particle. We expect that if we increase temperature, RNA will decay faster than the DNA. And by that ratio, we have a direct measure for, let's say, temperature history. We don't have a measure for actual temperature, but we have a measure on what thermal history have the particles gone through. So we put in the DNA and RNA into the particle without any thermal stress. We will be able to recover both of them. If we have thermal stress, the RNA will decay faster than the DNA. And experimentally, if we then take the ratio of the two and we increase temperature at a certain range, we see, let's say, a dependency of the concentration difference and the temperature. At the moment, still in a relatively narrow range. We are now working at increasing that temperature range. What else could you measure with DNA? DNA is sensitive to light. But for us, DNA by itself wasn't sensitive enough to light, so we added a small trick. This DNA here with the red arrow is caged, meaning that we have like a protection group we add to the end of the DNA, and if that protection group is on the DNA, it's not analyzed, it doesn't detect by PCR. So if the DNA is like this, we get zero signal by PCR. If we then shine light on it, we lose the detection group, and it's just DNA, and it amplifies by PCR. So this would give no PCR signal. This would give a PCR signal. We then encapsulate this DNA into our particles. We expose it to light, and we get a very nice correlation for light intensity shown on the particles and the difference of protected and non-protected DNA. We wanted to show that this can be used like a distributed sensor, so that you can sense at different points at the same time. And to be able to do that, we decided we want to measure the light shining on a cell. For a cell, we chose paramecia. So these are paramecia, small single-cellular organisms which react to light, but that's not so important for our experiment. So we decided, well, we'll take paramecia and we have them take up our particles. We've shown previously that various cell types can take up these small particles. You can see here in red, as we made the particles fluorescent, that in a fluorescent microscope you can see that they're taken up by the paramecia. Then we take single cells, pick them, and expose them to light. After that, we measure the amount of light by our, let's say, relative concentration assay, and we can see that irradiated by no sunlight, it's all still caged DNA. We can measure, and we can see at this is a measure from an individual cell, not aggregated from a sample. Taking one cell, dissolving it, and measuring the amount of DNA in the cell. Showing how, let's say, these numbers, or how being able to get a signal from these extremely low concentrations, how powerful that is. So by using this idea of stress, we can get temperature at the moment, temperature and light irradiation. We're working on increasing the scope of stresses we can detect. Another idea, just thought experiment, these 2 milligrams. So everything in this DNA world is in 1 milliliter and extremely small amounts, 2 milligrams, but that's 100 billion individual sensors, which we could distribute somewhere, and which we could go and say, well, we don't want to have just an individual temperature or light measurement, but we want to be able to do that measurement distributed over a whole field or of a whole area where we don't have access to. This technology, by the way, is currently being commercialized by 2 of my former PhD students. We formed the company Helixer, and we are mostly aiming in using this technology in product authentication, but also in underground reservoir characterization using DNA in particles as a novel tracer. So now I want to shift gears completely. I've showed you that we can use DNA in information, which is like a chemical idea of using information. But we kind of lost the engineering aspect of it to some extent. So this is like a more also personal story. Some of our papers received a lot of attention by media, especially the work on DNA data storage. Somehow media loves this idea of storing information in a genome. And so every paper that comes out, there was just one a few weeks ago, goes through all media. And we were kind of intrigued in finding out, well, how does that work? Why do certain papers appear in media? How fast does that work? What happens? Can we measure that? And can we understand that? So in the last few minutes that I'm going to show you, I'll show you kind of an analysis on how that happens through the eyes of an engineer. So the problem, or what we want to look at, is scientific news, which by itself, these sites have hundreds of thousand page views per day. They're extremely, extremely popular, not only by scientists. So let's have a look at the process of generation of a new scientific story. So somebody publishes a paper, and that goes out, published online, day 0. Then publishers or journalists or anybody in the internet can read it. And publishers and journalists will discuss with each other on what they will publish a news article. They will then write an article, and there'll be some feedback on the audience if they like it or not, and if the publisher or the journalist will write an article on a similar story. You will find out that most stories are related to DNA data storage or to food. A very famous paper is this one. If you don't know it, please look it up. Chocolate with high cocoa content as a weight loss accelerator. Was done by an journalist of the Science magazine, where he showed that he can publish something which sounds amazing, and even if it's wrong, the whole world will write media articles about it. So it's an important article in this world, but not the story I'm going to tell you. It's just taken as an example. What I want to say, it's a complex situation, getting from the original story to the news articles. And we are used to complex situations where we have some feed-in and extremely complex arrangement of unit operations, and we have some product out. And what we learned as chemical engineers is to use simple tools to abstract the complexity in our problem and use our very simple tools to describe what's happened. So let's try that for the case of the media generation. Let's say in representing it as a system, you have available online, you have like a Dirac delta paper published, and then you have this group of these online science journalisms. And they translate this original information into online news, which is then read by whoever in the public. And we would like to, let's say, understand this block of online science journalists. And this is best done if we can characterize the response, and then by the response and input, we can, let's say, characterize what this block does in the end. And there's a very elegant tool developed in the UK. It's called Altmetric. It's an online tool which measures news articles for every scientific article. So it's free data. For every article published, they generate a site which measures where it has appeared in public media. And so this is an example of different where that has appeared. We didn't take everything because there's all of food science, which generates enormous buzz. So we said we'll just take chemistry journals, and for every journal, we'll take the 20 most coveted articles. So those which got the strongest response, and you get about, from that, 2,000 news events. A relatively small data set, but that's what a data set that's freely available out there for everyone. So let's have a look what happens. How's the response of a single article? So this is the single most covered article in Nature Chemistry. So you see, one day after publishing online, 17 public news articles, and then it drops off to nothing after just 3 days. Another article, this one in Angewandte Chemie, you see kind of over a range of 20 days, you get the response. Another example, and if we take this extreme example, it takes 40 days where nothing happens after it's published online. And then after 5 days of it being in all media, it disappears into nothing again. So that's somehow was interesting for us. And one way to further go and analyze the data is say, well, let's say we have a time lag. until we get this response. Let's assume the following response. We have a time lag, and then we have a decay on how that response is. And we can analyze the time lag for all of the journals. We will see some journals, advanced materials, on average 30 days time lag between the moment the information is available and the day the first article is written on that. Strange somehow. Other journals, Nature Chemistry, same day as published online. The news appears, Angewandte Chemistry, also very quick. Then if we go and read the editorial policies of these journals, we see these published press releases the day it's published. So somehow there seems to be a connection between not the day the original article is published, but the day somebody publishes a press release. Either the journal itself, or perhaps a university decides, or department decides, oh, that's a nice story, we should have that covered in media, let's write a press release. And that might be responsible for this long time lag between the original information out there and the response we get. Let's look at the gain. Gain is something we would like to predict. I can't. So be able to predict how much response a certain article will get. But what we can do, we can analyze, well, what's the statistics, or what's the distribution of responses articles would get. And you see here on the log-log scale the distribution of responses that an article gets. And you see a typical heavy tail distribution, because you have a log normal, or kind of log normal decay of this distribution. If you know particle aggregation theory, particle aggregation gives results in log normal distributions, because a particle, as it grows, the larger it is, the more likely it is to hit another particle. So the larger it is, the faster it grows. And something very similar we see here, indicated by this log normal distribution, that we have like a aggregation effect. An article that already has news is more likely to generate more news, because the journalists, they don't read the original science. They read their competitors. What are they writing about? Well, we'll also write about that if we like the idea. And this is really kind of analytical proof that it works that way, or an indication at least that it works that way. Let's last look at the decay pattern. If we aggregate all of our articles, we see for all of the journals the decay pattern looks pretty similar. We can then further aggregate them all together. And most of you would say, well, it's exponential decay, first order system resulting in exponential decay. And if you plot that, that's pretty much what it looks like. However, we go and look at the data a little more carefully on the logarithmic scale, we see that we are missing a lot if we just assume that first order exponential decay. You then could go and think of any linear combination of systems which represent your system. We just said, well, if it's not first order one system, perhaps it's two first order systems. What does that give? And you can see that we then can nicely fit the data with these two. The number of parameters is slowly getting a problem, but it nicely describes our experimental observations. It also nicely describes our observations we had in reality with working with these journalists. Let's look at the system we come up with. We have a gain we can't describe, log normal. We have a time delay, two first order systems, which give us the result we get. Which is like this flow diagram, plug flow reactor, two CSTRs, gives exactly the same resonance time distribution as our system up here. So what happens? The time delay is the time it takes until a non-scientist writes a story. We somehow see by that nobody reads original literature, or journalists for certainly not. Somebody, or very rarely, and somebody after about 30 days decides that's something people are interested in, we should write a story about it. Then we have two different kinds of journalists. We have one which are really fast, so they have a response time of about a day, which is, if you consider 24 hours, them being somewhere in the world, that's extremely fast, and some which are really slow, a week. If you analyze them a little further, we find out these are just copycats. They just copy-paste the article they found somewhere and perhaps add a sentence or two. And these more or less generate more content. They get in touch with the authors and find out to get a little more background. Now we can use this analysis to measure the speed of information flow by using this first-order analysis. And there's a nice experiment from 2000. They published a link in a newspaper and then measured how quickly was the link taken up or read in the internet. And you can see that these half times are 3.5 days and 120 days for these two different groups of responses. If we go today, for example, the FIFA scandal, which was last year, the response to that is 16 hours for the fast and 1 week to the slow ones, which shows us how quickly information spread has accelerated over the last 15 years. It's a direct measure by this analysis as a system. First order, we can very quickly get, let's say, a measure on how fast information flow is and characterize it in the, let's say, relatively analytical way. So it's a game, somehow, for me at least, of using a chemical engineering tool to analyze something that has nothing to do with chemical engineering. It only analyzes people. But obviously, people can also be described, or the actions of people can probably also be described by similar systems as we use to describe reactors. I think important for us is that, one is that if we look at this science generation, that the story is the news. Nobody really cares about the science in the public measure. Don't get me wrong. People do care about science. With that small add-on to my work on DNA storage, I'll conclude that I hope I've showed you that DNA is an ultimate tool in chemistry to store and send information. The future of data storage most probably lies in DNA. It doesn't yet, so please back up your information. We can already use DNA as an information carrier in many engineering applications today. And I strongly believe that even if we only look at the information world out there, that the tools we have in chemical engineering are extremely useful in describing and analyzing information flows as they are traditionally used for analyzing product or liquid or gaseous flows. I would like to acknowledge 3 PhD students who did most of the work I showed you with me. This is the research group I share with Madeline Stark, working on this, many other topics as well. Public and corporate funding. I would like to thank you very much for your attention and happy to answer any questions.
I have a couple of questions. I found it very interesting. You talked about the link between chemistry and information, but your view was ultimately restricted on a relatively static view of information, whereas one of the most promising angles about information is dynamic information processing, how you choreograph it in time and space.
Yeah.
Would you like to comment on that?
Of course, I'll be very happy to comment on that. I think chemistry is, we have to take chemistry for what it is, what it can do. The things I've showed you are extremely slow. Hidden part of the things, doing DNA synthesis takes a week. So if you want to write information, that's... so in really generating significant amounts of information fast, at least with the tools we have today, we can't do that. On the other side, we have to think, well, there are fields where information has to be fast, let's say extremely fast and dynamic. But there are fields where it doesn't have to be fast. And we come from a world of books. Information in books is also extremely slow. And information in books in the 1500s, if you go to a monastery where we have a lot of information of those times from those books, that's extremely slowly generated information. So I don't think that everything has to be fast, and we have to find the spot where we start looking at combining information. And it can't do everything, for sure. It can't do wireless communication. There are many, many things we can't do with chemistry. But I think there are things it can do. It's very powerful. Storage is certainly one. We store information by physics, by, let's say, magnetic or optical, two physical states which are thermally very closely related. So that they're not very stable because they're closely related. If we go in terms of activation energy and induce that information into a chemical bond, which we have in DNA, we induce a lot more stability. So my thinking is more in terms of thermodynamics than really in having a fast enough access.
If I can just make one more comment. This was regarding your referencing of the communication theory literature like Reed-Solomon codes and so on. It strikes me that when you bring in chemistry and information, while you can draw on some of those tools in that literature, they are ultimately also based on very different abstractions. And it seems that actually you need quite a bit more new theory dedicated to chemistry and information and their intersection.
It's certainly true. If you want to build on this, you need more. What we did was really same as you have in a, so the coding is the same as you have in a DVD. We adjusted to some extent to, let's say, the limitations we have in DNA and adjusted a few screws to implement that. If you want to go further, and I think especially, so it's also a learning in both ways. So the chemical DNA synthesis world always wants to make perfect DNA because they want to make genes. This idea of having error correction, or that our whole world relies on the fact that we have extremely smart mathematical tools to correct for errors. That's not known in that world. So the moment I think it's more connecting the two things than generating more.
Thank you very much for a fascinating talk. A couple of related things. Just how long a piece of DNA can you store on one of these beads? You've obviously got multiple copies on them. You said you had 100 copies. How long can the pieces be? I mean, that's the error correction in itself for each bead you analyze.
So those are two questions. One is how long is the DNA? So in synthesis, the optimum is 150 at the root.
Yeah. How long of any DNA can you store on there? Could you archive people?
Yes. We can also store DNA. So we have a rather exotic project.
You get a chromosome on, or an entire genome?
Entire genome. So we have a project where we use the same encapsulation technology to store genomic information and add it to products. When you might think, well, when would you want to store your genomic information in a product?
A lot cheaper than liquid nitrogen for this year.
You can do it in liquid nitrogen, but you can also do it in silica. We've showed that we can store RNA, which is very difficult to store, extremely well, and it's extremely well preserved in our technology. So it goes much further than just the code. So we can also store, let's say, natural occurring DNA. We've done plasmids, genomes, various things which we've encapsulated.
Well, thank you for such a mind-expanding talk. My question's about weight. So I'm thinking, how much does information weigh? And if you work out the weight of the information, which is heavier, the binary code or the genetic code? So if I wanted to, for example, capture all the information that's on the planet now, how much would it weigh?
Yes.
And for example, I'm thinking if we wanted to send it into space,
yes,
and I wanted to use a rocket, I mean, weight's a key consideration in space travel. So what's the lightest way to send information?
So I would say for sure it's DNA. Because in binary, you always need a physical device to store it. So you have a magnet. You need a spin, and the spin needs a certain magnetic volume, or you need something measurable by a laser, 100 nanometers, micrometer size device which stores that binary information. In DNA, one single base of a 660 molecular weight stores 2 bits of information. And people have done, I've gone through it more or less, putting out the idea, all the information we have today, you can store in 1 kilogram of DNA. So that's
everything.
Everything. So there's also, in this world of DNA data storage, there's always an example that Facebook is currently building a data storage unit. I don't know how many square football acres, enormous. You can put all of that and everything else you have from Amazon everywhere in about a kilogram of DNA. That's kind of the metric it gives you. And if you, let's say, talk to funding, or that really relates to, okay, that's something really changes the way we think about information dramatically.
And I have one other question, if I might. You showed the two codes, the binary code and the genetic code. And it just struck me, if you could connect the two, then you would be able to connect humans to computers. And humans would become completely sentient with microprocessors. I mean, is that how close are we to that? Once you start thinking about, the brain is just an information storage device in a way, isn't it?
Well,
so why can't I link my brain to a computer?
Yeah,
and share my consciousness with you through a computer? Maybe because you don't want to know what's on my consciousness, primarily, I suppose. But
I think especially if we get further in sequencing, where that's more or less the connection between our genetic code and being able to put it into zeros and ones, then you get closer to that connection. Then going back is more difficult, because going back means that you have to induce genetic information into us. That would be gene therapy, which is possible, and is certainly how things are advancing. Not quickly that you can send me a message by translating genes to bits and then back to genes, but that we develop a therapy for a certain genetic disease, we synthesize the DNA, we get it into the body as gene therapy. So from an abstract view, that's possible at the moment, not from the futuristic view you put it in. But I think if you spin that idea further, those things will connect, if we want it or not.
Jeff, next.
So for applications that you want to tag, presumably you want the particles to adhere to the environment. So if you want a tracer, you want them perhaps not to adhere.
Yes.
So the surface chemistry is going to be quite important. Have you investigated that to any extent?
Yes, we have. We're working in various systems where we're looking at surface chemistry. And I think it's not only a challenge for what we do, because you want to be able to either have it, let's say if you're exploring rock, not to attach to the rock, but it's also a way to measure things. Because if you have particles where you can design that they're attached to a specific kind of rock, you pump them underground and they disappear, you kind of have a measure for that by the loss of the particles. And we've done surface chemistry. I mean, it's silica. It's pretty easy to do surface chemistry on them. Pegylation, there's a whole range. It has its own limits, of course, also in the stability thermally of the surface chemistry. But what we found out in tracing, let's say in the most tracer experiments we did so far, we get faster breakthrough curves with the particles than with traditional dyes, because the particles don't absorb on the solid phase. So there has to be learning on how these are applied in an environmental setting. And we have to do adaptations. So for example, rock works very well. Biological systems work very poorly with our tracers, because the particles foul and then sediment and get lost. So there we are still putting in a lot of effort in adjusting the surface chemistry.
Thanks.
You mentioned that one of the technologies that really made your whole field of research possible was rapid, low-cost sequencing. Really 10 years ago, all of your research was not feasible,
Completely
not possible.
useless, yes.
I think one of the amazing outcomes at the moment that has facilitated is personalised medicine. The fact that individual genomes can be rapidly and cheaply sequenced, and individual tissues and cancers and tumours can be sequenced, and personalised approaches to treatment can be done in real time or very short periods of time. Are there any applications of your technology, of your discoveries, to personalised medicine? Because I think there's a lot more money in personalised medicine than there is in data storage.
That's certainly true. I've never thought back, so turning it back again. So we steal something from medicine and use it for data storage. And then the question is, can I use that novelty that I generated back to the field of biomedicine? I really don't know. If you look at it more from a financial aspect, if you talk to a company in DNA synthesis, they see that in the biomedical world, they are hitting the wall, also for sequencing. Prices will not go below $1,000 because then the companies don't make any money anymore. There's no point in going lower than that. So these companies are now, let's say, hopping onto DNA data storage, thinking that this will further drive their technology in specific applications. So I think at the moment it's still going that direction. If we then can really, and that in the end would then lower personalized medicine cost if that would happen. As I said, we're still a big step away. It's possible, it works. It's, I think, the best way of storing information for a long time. But to make it, from an engineering standpoint, financially relevant, there's still a long way we have to go.
Thanks very much.
Time for one last question.
Thank you. Regarding the data storage, I can read a book of 500 years ago because Latin has been used as a lingua franca for 1,500 years, 2,500 years, 2,000 years. But probably the problem is not just the medium that you use for the data storage, is the language. I mean, probably I'm not able to open a file of MATLAB of 20 years ago. Yeah. How would you overcome this problem?
Yes, it's a really, really interesting topic to all data storage or to all data storage topics. And there are two answers to that. Firstly, so it's about interpretation. How do you best store information to be able to interpret it? And if you the moment you leave images, I think you're lost. Because you took the example of Latin language, I take the example of Egyptian, which for a long, long time we were not able to decipher Egyptian language until somebody found a stone which had the same text on ancient Greek and Egyptian, and then the connection could be made. So if you don't have information as pictures which can be directly interpreted, you have to have some kind of instructions on how to interpret that data. And those instructions have to be stored in a way that they're at least as stable as the way you store the information. And the second requirement is that they don't take a lot of space to store them. So you have to store them, let's say, if I can display my interpretation from digital to DNA, if you can put it on a stone or inscribe it in a gold plate, then I have that solution of interpretation. DNA has one advantage over many other storage devices, that will probably always be able to read it because it's our own natural language. So that can be overcome, but interpretation has to be solved. You have to show the person who wants to interpret it how it's decoded. That's a challenge. Interesting challenge, but not a chemical. Yeah.
So once more, let's show our appreciation for really an excellent talk. So,